Okay. Good morning, everyone. Well, good morning for me. Good afternoon to everyone online. So I want to talk today about developing in vitro models to guide oral peptide drug delivery, and a very important topic in the industry at the moment, as we're seeing more and more peptide-based drugs going towards phase 1 through phase 3 clinical trials. So it will give you an insight of approaches we're currently taking, from a formulation strategy, and then also what tools we're using to help guide those formulation strategies so that we can take it into phase 1 and then hopefully all the way through to onto the shelf. So to give you a background first, if you don't spend all day staring at the intestine like I do often. So the intestine, from the gastric epithelium all the way down towards the rectum, is a single epithelial layer thick. So when we're trying to deliver a drug, we just have to get across a single cell layer barrier, which is not a huge task when you think about it. But this is a simplified graphic of the intestine. So as you can see, it's got these finger-like projections that we call villus. So they increase the surface area. And then on top of these, you can see as you zoom in closer here, we have more of these folds. These are the microvilli that increase the surface area even more. So the goal of the intestine, as it is to, when we eat something, to have the largest surface area possible to get the most absorptive area. So getting across these with a drug is typically pretty straightforward. A lot of the drugs that we design, we typically will adhere to Lipinski's rule of 5 or the Pfizer rule of 5. So those are criteria in medicinal chemistry that I'm sure most of the medicinal chemists on the line would be aware of, where it has to adhere to certain criteria, normally factors 5, to allow something to be permissible to be orally available. And one of those is the fact that it has to be no more than 500 Daltons in size. And for it to be 500 Daltons in size, that means it can either pass paracellularly through the paracellular space in between the cells here, or it can be absorbed across the intestinal epithelium on the apical membrane here. So if you have a small molecule drug, this is normally very achievable. That's why so many of the drugs on the market that are oral are small molecules, and most of the biologics that we have are injectables, and the peptides tend to sit in the injectable space as well. So this is kind of how the cutoff we typically use. We say if it's 500 Daltons in size, and there's obviously wiggle room there. We have many drugs that come through to the clinic that are maybe 600 Daltons in size, but it really does depend on their physicochemical characteristics. And that really depends on how our medicinal chemists optimize those characteristics, so that we're able to make them orally bioavailable. And then we tend to look at the biopharmaceutics classification system of where it falls. So in the biopharmaceutics classification system, it relies on an XY axis of permeability versus solubility, and you want your drug to sit typically in highly permeable, highly soluble, and that one's very easy to deliver then. But then peptide drugs tend to fall in class 4, which is poorly soluble and poorly permeable, and they're considered poorly soluble in this case because generally the amount you possibly need, or they might be a BCS 3 where you have good solubility, but they're still low permeability because of their size. So when we look at a peptide drug that's maybe a 1 kilodalton to 2 kilodaltons in size, it's too physically large. So there's too much steric hindrance, there's too much shape and motility in the actual molecule itself for it to be able to pass through the paracellular space. And the reason we don't want to target our peptides typically to cross across transcellularly is because there's going to be a lot of enzymes present intracellularly. So there's a lot of intracellular peptidases, and their jobs are just to break down proteins and peptides into single amino acids or di and tripeptides so that they can use as building blocks in the bloodstream. So this is typically how food sources, so if you take in a high protein meal, your body's going to break down the majority of the proteins, but some of those proteins may be absorbed across the apical membrane, but they'll be broken down into free amino acids so that they can be used for building blocks of cells or proteins itself. So we have a lot stacked against us when we're trying to deliver a peptide drug, but we do know that peptide drugs are far more potent and powerful than small molecule drugs. We can use a far lesser amount of drug typically to get efficaciousness, to get efficacy. And then also we have much more targeted approaches with these peptide drugs. We know that there's less off-site effects, and they're very selective. Um, so when it comes to this intestinal barrier, the size exclusion aspect of it, because it's the biggest barrier to oral peptide delivery, we can overcome the gastric breakdown of the peptides by putting in either enzyme inhibitors. We can encapsulate it there's a multitude of encapsulation approaches, or we can use just typical formulation capsule or tableting approaches as well to protect it using polymers that will only be released once they get through to a higher pH around 6.5, which is what it is in the intestine. But the size exclusion aspect of the intestinal barrier is definitely the largest one, and it's pretty homogeneous across species. So the intestine is pretty consistent when it comes to absorption across different species. So we understand this hydrodynamic radii range pretty well. It's been characterized this is an intestinal cell line called CACO2 cells. They're originally derived from colon cancer patients, and they're an immortalized cell line that are considered the gold standard. So in the FDA bio waivers, if we want to get a drug through without having to do additional studies, we can seek out by proving its high permeability. And one of the ways to do that is by using CACO2 cells. So it's a really well characterized cell line. It is not the best cell line when it comes to mimicking the intestine, but it's one that is, we've done a lot of drug studies over the past 40 years or so. So it's very well understood, and we understand the hydrodynamic radii in those paracellular junctions due to that. And then here, when you look at rat, human, and dog, this is just using different size pegs in both of these studies here. So when you look at different polyethylene glycol sizes, you can see that there's kind of a steep cutoff around the 500 Dalton range here in both rat and human. But what's interesting is dogs, which are typically used in preclinical in vivo model, they have a cutoff, but it's much later. You see it's around 700 Daltons in size, and even at that, they still have a slightly leakier intestine. It's not fully understood why that would be. So dogs, although they're very regularly used for small molecule drugs, it is a question that when you're looking at peptide drugs, maybe dogs are not the best model to use. And that's for something like a pig, although pigs are much more cumbersome to use in the lab for numerous reasons. They have a much more predictive model of the intestine as humans. So it is a question when we're in the drug formulation, drug development space. So typically when a drug comes to me, it's a lead candidate drug. So that's the one that we're going to be bringing to the clinic. So then a question comes of which is the correct model for us to test our formulations in. And in this case, dog is not the most predictive. Rat, funny enough, in this case, it does become a bit more predictive when it looks at these. And then pig is probably the most predictive of them. And so this is just further characterizing that cell line that I talked about, the CACO2 cell line. So these are the range of small molecule drugs that have been tested in both humans. So on the right-hand side, this is an intestinal perfusion that was done in humans. This is the type of study that you would not see happening anymore. This was done in the early 90s at an academic institution. So I believe it was students were intubated with a hose down it through the gastric, and then they were the drugs were passed in, and then they measured it into the bloodstream. So they were able to characterize the fraction absorbed. So that's what we dose, the amount that gets past the intestine. That's what we're trying to figure out there. That's what the fraction absorbed is, and we were able to correlate that against the human permeability factor, so the PF. And then if you look at it the same in the CACO2 cells, when you look at the fraction absorbed in humans versus the PF, so PF is the in vitro equivalent of PF here. You can see that these map very well to each other. So we know that this cell line is typically very good for predicting this when you have high permeability drugs. So that's the thing. So it's a great cell model when you have things that are well permeable. And when we're talking about something that's highly permeable, we're talking about things that are anywhere between 40% fraction absorbed and higher. So in the small molecule drug space, that would not be considered good. So if we were trying to work on a atenolol nowadays, would not be the best drug to work on. So peptide drugs tend to sit in this space down here. They tend to have permeability values around 10 to the minus 8, 10 to the minus 7 due to their size. So when you look at these models, you can see that it's not going to be the most predictive. So the big question is, number one, is the model correct? And then how do we predict some of these formulation choices or even improve some of these in vitro before we go to in vivo and even into the clinic if our models aren't the most appropriate? So as I said, um, peptides are hard to get across. So one approach to use is using intestinal permeation enhancers. So intestinal permeation enhancers are have been really well studied in academia, and some of them have successfully made it to the market that I'll go over in a moment. But typically, you get medium chain fatty acids tend to be the largest class of permeation enhancers that have been well researched, the most well researched class of permeation enhancers. But you do also get these novel small molecule ones like phenylpiperazines, which are, if somebody's a chemist, they'll know that it's kind of the structure of a phenylpiperazine. It's two cyclic structures bound together. And then there's also some more novel ones that are antioxidant type ones. And then you have derivatives such as trimethylated chitosan chloride, which is a polymer-based permeation enhancer, and then the dodecyl maltosides, which are fatty acid derivatives effectively as well. So all of these work in part by interrupting that paracellular space and pulling those tight junctions apart for a short period of time. And it's what happens in our body anyway. So when we're consuming large amounts of fatty foods, that will happen for a short period of time. It tends to be a period of about 15 to 30 minutes where the tight junctions are perturbed. But there is also a size cutoff. They're not just open so that bacteria and LPS can flux across. And this is a question that's often asked. We do know that generally we see as it goes above a certain threshold of about 5 kilodaltons, we tend to see a substantial decrease in the amount of permeability that is present there. So something that's LPS, lipopolysaccharide from bacterial cells, which would cause a inflammatory response, it's very unlikely that any of that will be able to go through because of the large size there. So these medium chain fatty acid permeation enhancers. So as I said, pretty well characterized. So I've got three on screen for you here. So this is sodium caprolate, sodium caprate, and then salcoprosate sodium, or SNAC. So the three of these have made it into clinical trials and shown pretty promising results for various different companies. So the top one here, sodium caprolate. So that's a medium chain fatty acid, sodium salt of it, and it's the predominant medium chain fatty acid and permeation enhancer in chiasma's oral octreotide formulation. So octreotide is a peptide used for the treatment of acromegaly, or colloquially known as giantism. So it's a growth hormone instability in the body. So this is the first oral octreotide formulation that made it to the market. So commercially came onto the market, I believe, in early 2020, maybe late 2019. So that was the second or third peptide drug that made it successfully. So when we talk about these peptide drugs, the one that we're not talking about is cyclosporin, which has been on the market for a good few years, but it breaks the rules of oral peptide because it actually crosses transcellularly, and nobody typically wants to design a formulation like that. So when we refer to these, we're excluding cyclosporin from that conversation. And then the second one here, sodium caprate, very similar in structure, just two carbon chain lengths longer. And this in vitro and in animal studies and also in the clinic is shown to be more efficacious. As you increase the chain length of these medium chain fatty acids, you get increased efficacy, but then you get decrease in solubility. So C8 through C12 are kind of the sweet spot where you're able to get solubility. If you go higher than that, you definitely do struggle with solubility, but you do get increased efficacy. So sodium caprate was originally licensed by Marion Pharmaceutics and their GuyPet formulation. So that's the gastrointestinal permeation enhancing technology formulation. And that was licensed by Novo Nordisk for their oral insulin programs and also their oral GLP-1 analog programs. But it was terminated when Novo Nordisk decided to move on to SNAC, which is the next one here. So SNAC is a medium chain fatty acid derivative, here as you can see, of salicylic acid. And this was originally Emisphere's Elegant technology. So you'll see that a lot of these permeation enhancers had other companies, formulation-based companies typically, who developed the technology, and then other companies would license it, or in the case of Novo Nordisk, who licensed it and then eventually bought this technology. So they used this for their Ribelsis formulation, which came on market around 2019 also. So it was kind of gangbuster year for oral peptides. It was very exciting for some. I did my PhD in oral peptide delivery, and I've worked in the space since. So it was really exciting to see some of these products coming onto the market. So Ribelsis, which you may see, it's quite common in the news at the moment. It is primarily used for diabetes, so it is an oral GLP-1 analog of semaglutide. And if you've seen, it also appears to be a weight loss drug, so you'll be seeing that quite a lot. There is a shortage of this drug in general, but it's not Ribelsis, it's their actual partner injectable drug, which is the semaglutide there. And then another typical permeation enhancing technology that is often used is Labrazole ALF from gas phosphate. So this is a oil lipid-based formulation. So these three are normally used in solid dosage form. The chiasma formulation is an oily liquid, and Labrazole is also kind of this fatty acid based oil. So it's both PEG and glycerol esters of these fatty acids here. So again, you're just seeing this similar motif found across all of these permeation enhancing technologies. And Labrazole is typically, and I didn't describe it here because obviously it's a mixture of a lot of different PEGs and esters here. But predominantly C8 and C10. And Labrazole and Gafas, they have a couple other ones. So they have a solid dosage form called Chalosir, which is similar in formulation. And they're typically used as solubilizing agents, is what they're marketed as, because they will improve the solubility of lipophilic drugs. But they're also been shown clinically and preclinically to be intestinal permeation enhancers. But we are particularly interested in Merck in sodium caprate because there was some really exciting news over the last couple of months that we published our phase 2B clinical trial using sodium caprate with an oral PCSK9 peptide. So this is a brief snapshot of some of the pharmacokinetics of it. And so the peptide itself is about 1,500 Daltons in size, so typically wouldn't be very orally bioavailable. And the abstract for this first was in 2021, I believe, where we presented at the American Heart Association of our phase 1 clinical trial. And then this is a snapshot from a paper that's coming out. So it's just been released in circulation of our phase 2 clinical trial of our single ascending and multiple ascending dose, and then also the efficacy studies as well for our PCSK9 peptide. So it's for dyslipidemia for our cardiovascular program. But what's interesting is that we were using sodium caprate with this as an intestinal permeation enhancer. And we noticed some issues in the clinic regarding formulation aspects. So as you can see here, so this is when it's 20 mg or 10 mg of the drug. And then it's 360 or 180 mg of the sodium caprate. So as I said before, these peptide drugs are very potent, so we're able to use really small amounts of them. But to help them be absorbed, we do need to use this permeation enhancer. So as you can see, when we use the 20 mg with the 360, we get, so this is the trough MK616, which is the name of the peptide, plasma concentration. So this isn't a typical PK curve. So this is the actual C trough at the end of the day. So you can see that as we're taking it in sync conditions, so taking an oral formulation daily, you can see that the amount that's in the systemic circulation is increasing, and then it kind of stabilizes around day 7, which is kind of what we want. We want to be able to control the lipid content so that people who are at risk of cardiovascular events will have a much better quality of life and less risk factors there. But we also looked at decreasing the sodium caprate concentration of understanding and the dose. So here we dropped down 10 mg of the actual dose, and here we dropped down 10 mg and halved the sodium caprate concentration. And we found that this is kind of the sweet spot that we're able to live in, that 180 to 360 mg of sodium caprate, it doesn't seem to, this is kind of our cutoff. When we go lower than this, I think from our previous abstracts that was in American Heart Association we looked at 90 mg as well, and it wasn't successful. We saw concentration dependency. So we think that for this peptide in particular, 180 mg is sufficient for us to get clinical efficacy. But one of the big things we noticed was that once there was a fed state, so these were all fasted, but when there was food involved, so there was postprandial, so we gave the formulation after 30 minutes after a meal, we saw a huge decrease in the efficacy and the pharmacokinetic profile. So that suggested that there was something at play here with our permeation enhancers and food effects. And the problem was we didn't have appropriate models to look at that. So we had some preclinical models, but as we discussed, the dog is not the best one for peptides. Rats, when it comes to feeding, they're not the most typical because they don't secrete bile the same way most higher order animals do. And then pigs are a very cumbersome model. So if we're trying to tease this out preclinically to try to guide our formulation, we want to be using as little material as possible. And if we're using dogs, they're quite large animals, so we'd be having to use quite a lot of material. So then it comes the question of, can we take some of our in vitro models and make them better so we can look at these types of questions? And kind of that's where the research that I lead at Merck in particular, so I support and I lead the development of the in vitro models to support our oral peptide programs. So there's two main thrusts that you can use to improve some of these in vitro model predictions. So one is looking at changing the cell source, so moving away from just the plain CACO2 cells, moving towards intestinal organoids, which are, we know, more predictive of in vivo biology. And then also looking at different models. So the traditional model that we use is called a transwell model, which is a cup inside a well. So it's a static model, and some of these microfluidic devices are dynamic flow models, which are, again, more representative of in vivo biology. So looking at these types of things and combining them with in silico modeling, we can maybe get much better predictions and guide our formulations a lot better. So this is a very big splash up of kind of a lot of the approaches you can take. So as I said, this is what we would typically see in vivo. So we have, our intestinal buffer or intestinal fluid here, then we have mucus, then we have our cells, and then, we want to look at the drug coming across. But we don't have these models typically in vitro. We tend to look at just this transwell model that I said, which is just cell culture media on both sides, and then just the cell line here. We can apply some mucus there, but it isn't the most representative. There's these goblet cells, which are a CACO2 derivative or HT29 derivative, which produce mucus again, but it's not the most predictive. So all in all, these are all the things that we would need to encompass to get these models more predictive. But then the question is, do we need all of those things as well? And often for looking at things like for some of these formulation guiding approaches, we don't need all of the additional complexity, such as the immune cell. Maybe we need mucus, and the buffers are a very important one. Cell culture does not represent the complexity of fasted state or fed state in simulated intestinal fluid. And as we saw, we did have that decrease in efficacy when there was a fed state there, and we're not certain why it was. Was it there's a multitude of reasons that could be happening there, and we're not certain what it could be. So one approach is to use intestinal organoids in the transwell. So taking the typical conventional model, but changing the cell source that's present there. So instead of using the CACO2 cells that we typically use, switching them out for intestinal organoids, which are more reflective of in vivo biology. So the problem is then you splinter off, and now you have two options. You have, well, three technically. You have iPSC, ESC, or biopsy-derived organoids, and each of them are slightly different. So in this case, we have induced pluripotent stem cell-derived intestinal organoids. So this is where we have an induced pluripotent stem cell, and we can direct it to differentiate into the cell or the cell type we want. And in this case, it's small intestinal and colonic epithelium. And you can see from this, when we were looking at the CACO2 cells versus the colonic and small intestinal organoid, the apparent permeability coefficient, so that's the piece that I was talking about earlier about how much of the drug is able to get across that membrane, decreases in order here. And we do know that this is the case in vivo. We know that the small intestinal tissue is much more leaky than the colonic tissue. And that makes sense. In the small intestine, you're trying to get increased absorption of all those nutrients and peptides that are present, whereas in the colon, you have a much thicker mucus, you have lower motility, and you also have a lot of bacteria and excretory factors, so the metabolites that are trying to be excreted from the body. So in general, in the body, you don't want those to be absorbed. So the colonic epithelium is much tighter or harder to be absorbed across. And we found that when we differentiated these iPSCs into the two different tissue types, we did see a decrease in permeability here. But notably, the CACO2 cells were much less permeable than the colonic organoids as well. And this is a known fact, and that's where it brings you back to kind of it's not the most predictive for poorly permeable drugs. So, this suggests that this type of model can be effective. As you can see here, it doesn't grow as a flat surface like we typically have depicted here. In CACO2 cells, when you see them onto a transwell, they will grow flat, but in organoids in general tend to retain this somewhat convoluted structure. So you see that there's kind of pits and troughs because they like to grow in these different structures because we typically grow them that way, and it kind of retain that memory as well. So then another approach that I mentioned is instead of switching out the cell source, you keep the cell source the same. So this is going to be CACO2 cells here, but you change the model. And in this case, it's a microfluidic device, so a good on a chip model. So in these models, they tend to be pretty small, and the chamber itself is about 70 microns high, and the width is about 14 microns wide, but obviously they vary from company to company. But that's what this one is that we also use at Merck. So in this case, you can see how, it's not the best microscopy image, but this shows you the structure that they kind of grow in. So you see the cells on this membrane, and as the flow is running both bilaterally on the apical and basolateral chamber, it causes what they refer to as spontaneous differentiation. I don't believe that is the correct term, but it causes a kind of a fluidic shear stress environment that causes the cells to retain that memory of its original structure, and it tends to grow in these convoluted structures. So as you can see, as opposed to growing flat, it tends to grow up in these villus-like structures that we know were present in the intestine originally. So you can see here when they do a cross-section imaging that you can see kind of this, the cell nucleus are here. This is the villi is stained here in green. And then it's kind of like a hollow tube here where there's just no real growth there because they can't access the nutrients as well. So good on a chip kind of came out in the early 2010s and has been something that industry and pharma are interested in, seeing how we can use this for beyond typically disease in a dish models, but also how can we use these for looking at formulations and guiding our understanding there, because it is a much more dynamic system than a typical transwell model. The problem is, so they used CACO2 cells, as I said. So this is a different paper from a similar kind of thing, where they were looking at the RNA transcriptome. So this is just a PC plot where it shows the similarity or dissimilarity of something. So if things are close together, it means they're transcriptomically similar. So you can see that jejunum and duodenum and the odd ileum from these donors seem to be very similar transcriptomically. Makes sense. We know that a lot of the transporters, a lot of the growth factors are quite similar in these tissues, but colon is quite different. And we know that that's pretty well established. And then when you look at CACO2 cells grown on a transwell or CACO2 cells grown on a chip, they are very different from each other. So the expression factors and the transporters expression changes quite a lot here. But the most interesting thing here is that they're not similar to native tissue. And that is a problem. So we can see that by modifying the model, we get a difference in expression profiles and transcriptomics. But if you're still using that cell source, we're still going to have issues of consolidating it towards actual in vivo biology. So it suggests that rather than having to change both the model and the cell source, we need to change both of those things, actually, if we really want to get the most predictive model. So this is another example of how the actual gut on a chip looks like. So when we grow the cells on it, so this is induced pluripotent stem cell organoids. You can see that these are the villus-like projections that we talked about here. And you get the cell nucleus in is in red here, and then we have the villain on the outside is green. So that just shows the apical membrane. And we were able to characterize this type of model that it is physiologically responsive. So when you treat it with TNF alpha and interferon gamma, we get an increase in the amount of permeability of that labile molecule that's about 3 kilodaltons in size, but we don't get a decrease in viability. So it suggests that they are pharmacologically or reactive to stimuli, which we would expect to see in vivo. So then this is a paper I love. I was not involved in it in any way, shape, or form, and they kind of beat me to the punch somewhat for some of the stuff we're doing at Merck, but it's a really great paper, so all dues to them. So they were looking at a simultaneous flow and apical and basolateral, which we've found it's been shown in previous papers. That's where you get the most differentiation, or that the most efficacy from those cells. And they were looking at using transwells versus the chip, which is something we're also characterizing because it's really important to standardize it back to what is the gold standard. And they were looking at the permeability of mannitol, FT4, so that's the 50 dextrin 4 kilodalton. So it's a very commonly used molecule in oral peptide delivery because it's a fluorescent dextrin of around the same size as the peptides we look at, but we're able to assay it very quickly. And then insulin, which is also a commonly used one in this space, because again, we're able to measure it pretty quickly. And they were looking at it in transwells or in their chip, and they were looking at it without the presence of a permeation enhancer or in the presence of a permeation enhancer. So this is tetradodecyl maltoside. So it's another one of those fatty acid derived sugar type permeation enhancers. So as you can see here, the one thing between the transwell and the chip is that they observe this, and we also see this in-house, is that in the chip, across all groups here, you see that the basal permeability, so the amount of drug that fluxes the same cell source, so I believe they use TACO2 cells here, in the same cell source in a chip, that the permeability coefficient was higher. And we see this as well. So you can see that it's about twofold higher in a couple of these here, and we see the same in our hands. And it's believed that the TACO2 cells in transwells do have too low of a permeability coefficient. And we normally have to apply a multiplier or a fudge factor when we're calculating these things, comparing it to the in vitro in vivo correlation here. But what you can see is that this chip has a higher permeability, so it may actually fall closer to in vivo effective permeability coefficient. So that's exciting, and we're seeing the same thing coming out of multiple groups, that this is kind of a consistent piece when it's grown in the chip with that microfluidic flow. It seems to be more permeable and more reflective of biology. And then when they put in these permeation enhancers, the tetradodecyl maltoside, you see that they saw similar efficacy in both the chip and the transwell, but in most cases, in these two cases, that they did see increased efficacy of the permeation enhancer in the transwell compared to the chip. This one questionable because really large error bars. And so it seems to be a trend that the permeation enhancers seem to be more efficacious, and maybe falsely more efficacious in the transwell model, which makes sense because you're doing direct exposure and there's no escape for the cells in that regard, as opposed to a flow condition. There's increased surface area, but there's also flow, so the actual exposure of the permeation enhancer is lesser to the cells. And we see this similar in our hands, so I'm going to give you kind of an overview, but just to kind of give you an idea of what this looks like. So this is the Emulate chip system. So Emulate are a company that's spun out of the Wyss Institute. So the Wyss Institute were the original producers of this paper, and they kind of developed these model systems. So Emulate is a company that we purchased their equipment, and I worked with them during my postdoc as well. So when I came to Merck, it was quick for me to get this set up. So it's a system where we have to, it's a PDMS-based chip. So we have to functionalize the PDMS to allow the cells to stick to, otherwise the cells won't be able to adhere to them. So we functionalize the PDMS and coat it with Matrigel to enable the cells to stick. We seed the apical chamber here, the blue chamber, and then leave that overnight. And then the following day, we'll attach this pod. So this is the pod and this is the chip. And the pod is where we'll have our media for both the basolateral and apical. In this case, currently, we're just using one cell type in the apical chamber, so we just put in the same media in all of the chambers, and then we normally culture that for about 8 days. And then we're able to run our assays. So initially, when we got this system, Emulate has characterized with CACO2 cells using the BBBE, which is their brush border enterocyte subclone of the parental HCB CACO2 cell line. So we had to go back and recharacterize the system using the HCB, because this is the one that's more approved, is the approved cell line for the FDA. So as you can see, we measured the basal permeability over time. So we seed at day 0, and then we're just measuring the basal permeability over time by using Cascade Blue Dextran 3, because it's pretty detectable in the media. So we flux that over time, and you can see between both cell lines, although the CACO2 BBE is a bit more noisy, they're both pretty consistent. There's a tight barrier in the 10 to the minus 7 range, which suggests good barrier integrity. But it was great to see that the cell line we were more interested in had much greater consistency, and this was over the course of about 10 or so replicates. So taking that forward, we then were looking at how consistent this is over. So each of these gray colors is about 12 chips per group, which is why there's so much error. So this is the standard deviation. So then the cumulative values here are about n of 70, because we really wanted to characterize the basal permeability in all the studies that we're doing with this. We want to make sure in industry that it's a very robust system, so that when we're testing anything, that we know that we get consistent results. And I think that's one of the most important pieces about industrial assay development, is that the robustness is there in particular. So when we look at the basal permeability, we see it's pretty consistent from about day 6 out to day 15. So it falls in around between 1 and 2 by 10 to the minus 7 for this Cascade Blue Dextran. So that's where we tend to do our assays in this time frame, looking at both small molecule and peptide drugs to validate this. So we're also comparing transwells to chips, similar to that previous paper that I talked about. We're looking at sodium caprate, which is one of the permeation enhancers, as you saw earlier, was in our clinical trial with our PCSK9 peptide, and then also sucrose monolauride. And the reason for that is it's a pretty well characterized permeation enhancer. It's not the best for a oral solid dosage form formulation, because it is a sugar-based permeation enhancer. And unlike the tetradodecel, dodecel multicide, sucrose monolauride becomes quite runny at low temperature. So around 60 degrees Celsius is a liquid. So for forming capsules or tablets, it's quite difficult to work with. But what we're looking at was the concentration dependency in both our chip and our transwell model. And you can see that as we increase, we get, this is in 10 log here, just for ease of data interpretation. We can see that around 10 millimolar for sodium caprate and about 1 millimolar for sucrose monolauride is kind of like that sweet spot of efficacy without going too high, that we know that we're probably killing the cells in a CACO2 transwell model. But then when we look at those same concentrations in a chip model, they're doing nothing. So you're getting a 0-fold increase, and you're getting maybe like, I think it was 1.2-fold increase on the 1 millimolar sucrose monolauride. And we found that we looked at a range of concentrations. So there were higher concentrations as well, but we believe they were also killing the cells. So it looked like 25 millimolar and 10 millimolar sucrose monolauride and sodium caprate, respectively, were the efficacious concentrations here. So to us, we're doing a lot more that you'll see coming out. Hopefully, we have a manuscript that we're submitting on the full characterization of this model, but unfortunately, it has not been OSTIC cleared, which means we can't present it externally yet. So this is kind of a teaser of kind of the stuff we're doing. So we're just robustly validating this model with both small molecule drugs, so to correlate back to the PF that we know in human and the fraction absorbed, and also using some peptide drugs in this system with permeation enhancers to show how predictive this is. And we're also looking at trying to replicate those food effects that we saw in the clinic. So overall, that's kind of what we're doing at Merck and across the board in other companies to develop in vitro tools to guide oral peptide drug formulations. And so with that, I am happy to